try to start now with a presentation. And my question now is, do you see it in presentation mode? Welcome everybody to the, it's always interrupted by the recording in progress, recording started and so on. So now I start again. Welcome everybody to the second international project workshop. And this is the welcome to the open session at day 2. We already had a day 1 where we had some meetings with the stakeholders. In specific cases, these were the stakeholders of industry and regulators, where we will hear about within the second half of the day today. And there will be a third day, something after today, where we will have some further expert sessions. So this is day 2 of it, and I welcome everybody to it. And nicely, Klaus already introduced you to the housekeeping rules here. Just a quick reminder to those who just entered, we are up to 200 registered participants. 85, as Klaus said, already joined. Please keep yourself muted during the presentations so that everybody can listen easily to the presenters. Use the chat function and the question or Q at the beginning so that we really see and clearly identified easily in the chat if there is a questions. So additional comments, suggestions, and questions are really very much welcome, either here in the chat function or specifically also in a mail after the workshop. And the recording was already mentioned. So when we think back about a year ago, it was also in the beginning of November, we had the first workshop of NanoHarmony with gap analysis, and to introduce to you all the activities with regard to guidance documents and test guidelines. And the first one was really extremely well received. And this was something which we found very important, which was really helping us to do our job during the next year. Even though during Corona, certainly there were some downs with all the motivation. This very much helped us to move on and to push forward, because it showed us also the international interest in the activity we are doing in the safety assessment of nanomaterials. So this is an important issue, and the harmonization on OECD level, especially the test guideline and guidance document development. And over the last year, there were several webinars, and over the last years, there were several activities from expert meetings. And the expert meetings now today, yesterday and tomorrow. And what we do here today is really then giving a summary, again, to all of you of what are the activities and what's going on. This international collaboration, which is facilitated by these types of workshop we are having here with the NanoHarmony workshop, is really something very much appreciated and needed really to achieve this international collaboration and to achieve the test guideline. It also facilitates and help much, it is also helped much and facilitated by OECD activities and the EU project Nanomet and Mar Gonzalez pushing forward this kind of collaboration. And with this, I really would like to thank also for this collaboration here. And the results of the collaborations are seen yesterday and tomorrow and during the open session today. NanoHarmony is one of the projects related to the Malta Initiative. And the Malta Initiative has the main aim in looking how can regulation keep pace with innovation. And looking into how can we keep pace the harmonization and harmonized testing and assessment of the safety of nanomaterials is one important issue. And this slide, I think most of you have seen it more than once. So it really quite easily shows on the left-hand side the different types of inputs we are using within the NanoHarmony consortium, and along with the associated partners, which we very, very much, and I would like to thank the associated partners also very much for contributing and collaborating here with NanoHarmony and the Malta Initiative. It's on the left-hand side, the input, which is coming from the Malta Initiative, from National Initiative, from the Nano Safety Cluster. We have stakeholders from industry, from science, from member states, and from regulations, and from the Commission. We have regulatory bodies and associated or agencies associated in this relation to regulation, like ECHA, EFSA, EMA, non-European parts as well. We have the OECD as a main standardization body, but also collaborate as much as possible with ISO and CEN. And available data, methods, and information is one of the bases of NanoHarmony. We are asked, and we are currently using as much as possible already available data being produced in all the national and international and European projects. So this is done, and this is the input. We have then the 3 work packages, and the way forward is here, certainly the part where we achieve and work on new or adopted OECD test guidelines and guidance document. We have the aim of improved cooperation between scientists, industry, standardization bodies, and regulators. And another aim, the way forward, looking into faster implementation of scientific results into regulation. And just showing a little bit here, we want to go from research to regulation and certainly facilitate the development and the use of the legislation. And we have 2 major aims or 2 major points we are working on today. Certainly in the middle, the formation of networks. This workshop is part of forming this kind of network with all relevant stakeholders. And we have in the morning sessions the focus on the development and on the validation of test methods for OECD test guidelines and guidance document. And we have in the afternoon really then the focus more related to the translation of science into test methods and to speed up this kind of process. And these are the 2 foci we have within this workshop today. And especially the latter one wasn't a focus in the first workshop, but is one focus during this workshop. So with the aims of the session for today, we can summarize it. Certainly we want to bridge science and harmonize testing for regulatory purposes. So you please keep this in mind when listening to the different presentation, to the different sessions, and please help us that we can really pursue these aims and really achieve these aims. We want to inform you about the ongoing and the developments of the test guidelines. We want to present the outcome of the workshop stakeholder session, which were held yesterday in the morning and in the evening. I would like to mention this specifically because to get all stakeholders involved, we wanted to reach out internationally. So we had the same sessions in the morning and in the afternoon, Central European time, to enable and to make it a little bit more easy to our colleagues, international colleagues in Asia, and to our colleagues in the US and all the other continents to work in these and contribute to these stakeholder sessions for industry and regulators. Intensify cross discussions will be one of the discussion points this afternoon and facilitate the international participation in the test method developments, inter-laboratory tests, and so on. So coming to the point, you will see here on the left-hand side, the sessions planned for this morning from 9 with my introduction leading up to 1 o'clock, which is then the last one by Maria Duzinska on the update on risk-on developments. And what we did for this morning is to present you an update in brief, very brief, of what's ongoing with regard to the different test guidelines to make it public and to make it available to everybody. So on physical chem properties, on health effects and effects on biotic system is another session, environmental fate and behavior, and the updates on the Malta initiative and other test guideline developments. The afternoon, as you can see here, we start again. We only have a half-an-hour lunch break, so prepare yourself for a quick lunch. And we will have some discussion of the morning hours in the beginning, and then we have the look into some other projects and their results, as well as, as well as a wrap up of what has been discussed yesterday, the feedback and discussion of the industry and regulators session, which I'm very much looking forward to see on this interaction, as well as identifying barriers between science and regulatory tools. So with that, I think we look forward to a very intense, full calendar agenda for today. And just to not start with too much of a delay, I pass on the word to Klaus Svensson and wish you all a fruitful discussion and a lot of new information and a good day.
Thank you very much, Thomas. If you can stop sharing your slide, then we can get Jacques to put his up.
I have to somehow get.
It's at the top. Well, somewhere
on the top right. Thank you. Now it's stopped.
Thank you, Thomas. And we're now at 103 people. So no pressure, Jacques. If you want to start sharing your slides, so we can get into the physical chemical properties session.
Yes, hello everyone. Yeah, I hope. Can you see anything?
Not yet. No. Here it comes.
Here it comes. Okay, let's see. I put that in a mode. Let's see here. Yeah, that's so many things here. Okay. You see the presentation mode now? Yeah.
Yeah, but it's coming.
Okay, correct. Okay. So okay, I'm Jacques Bouillard from INERIS, France. Do you see anything?
Yeah, it's in the. If you go to the parameters dysatrophy, the menu at the top left to the second menu, it's in the wrong slide mode. So if you go right to the top, you should have a menu that says parameters. You don't see that. It's sharing the wrong screen basically.
Okay.
If you go, if you unshare again, sorry. If you stop sharing.
Paramètres d'affichage.
Yeah, I don't think Jacques can see it. It sometimes happens they can't see it.
No, I don't see it. Anything.
Okay, so if you stop sharing.
Oh yeah, here it is.
Yeah,
okay. I got it here.
And then just switch them. Yeah.
Yeah. Okay.
Perfect.
Is it better now? Okay, I'm sorry.
No, don't worry.
Okay, thank you, and happy Thanksgiving to Americans anyway. So I'm going to present test guidelines on dustiness and test guidelines and guidance document. Thank you. I'll thank also the collaborators, Alexis Vigne, Anya, Anna, Sofia Fonseca, Carla Ribalta, Keld Jensen from NRCWE, and Anna Paul and Elizabeth Nisch from BAuA. Okay. So what is dustiness? Why is dustiness important? Because dustiness is a critical element for exposure risk analysis and ETEX risk analysis. But to carry on the dustiness study, we need standardized dustiness test applicable to nanomaterials and necessary to support the current legislation and future legislation. So two projects are going on now to carry out this work. Go4Nano and NanoHarmony that provide the scientific basis for dustiness testing. Partners are, of course, INERIS and NRCWE, BAuA's leaders, and we have also international partners, cooperating partners, about 20 on this project. So what's the aim of these OECD test guidelines and the guidance document development? The project is hinged on the three phases. First, the development of test guidelines on dustiness testing for granular and high aspect ratio nanomaterials. We call also RN materials. That would describe the comparison of different dustiness test methods, namely mass and number-based dustiness indices, particle emission rates, particle size distribution, and so forth, to facilitate the use of this dustiness data for exposure assessment. The powders that are targeted are basically spherical or granular powders and fibers. And we also will describe these method-specific ranking powders on dustiness categories. For example, a powder could be of very low, moderate, or high dustiness. And this will be very useful for assessment of release potential, namely in safety by design type of approaches. And also, this TEG will provide elements on groupings and read across on dustiness level for powders that have similar characteristics and emission potentials. First phase. Second phase is how do we use these data for exposure assessment modeling? So this could be part of a GD guidance document for this, and in which we'll explain what is the activity modifier, or also the energy factor that is also the same name that we gave to. So, and we'll use these source terms that include these dustiness data indices to provide a concept of how you use this data to do some exposure assessment, modeling of exposure assessment, and to do some exposure risk at the workplace. At the same time, so there another GDE, how do we use dustiness data for explosion risk assessment, ATEX risk, and basically by investigating relationship between dustiness and explosivity. And also this modeling that they also involved in the previous GD will be used for describing different accidental scenarios and to provide some ATEX safety management approaches. So here, very quickly, the project, of course, one part is from help with Gov4Nano, the other one with NanoHarmony. Gov4Nano are aiming at the granular powders, while NanoHarmony is aiming towards HAN, high aspect ratio fibers, let's say. So those are different type of powders that are treated. Different also aerosol generation methods that are listed here for the different project, sub-project. Different measurement analysis that are listed here. I won't go into the details, but the end effect is to use all this data for modeling exposure as a GDE, and application also in explosion risk also, and developing method-specific ranking schemes. So in terms of timeline, we are now working on the development of these tests and doing the testing interlab and intra-lab comparisons. So hopefully in 2022 we'll be able, end of 2022 we'll be able to have the basic data, and writing of the TG GDs in 2023 and submission in 2024. So first on work for the TG on non-HARN dustiness. So the objective here is to provide the generation of dustiness data and development of method-specific dustiness ranking scheme for classification, future classification of nanopowders. So here you have different methods, generation methods, aerosol generation methods of different energy that are tested: the rotating drum, continuous drop, small rotating drum, a vortex shaker, fluidizer, Venturi devices, and so forth. And we also try to work on 6 powders, the basically TiO2, SiO2 that is listed on the screen here, and with the 7 method that I just described, with a high priority on methods for which we have more than 3 laboratories that are involved, namely rotating drum method, the small rotating drum method, and the vortex shaker. So, so the idea here is, of course, as interlab, I mean, these TG testing involve interlab test, intralab test, and use also some established methods, namely some methods that are already standardized, professionalized. So the impact are basically twofold. First, a regulatory impact, basically by gearing towards the current and foreseeable regulatory needs, of which Annex 6. Another impact is the use of this data for the occupational risk assessment and ATEX risk assessment. and also the use of that data for method-specific ranking, dustiness ranking, and grouping and read-across dustiness levels that could be used for similar characteristics or emission of powders. This now for the dustiness of ARN. ARN is a high aspect ratio nanomaterials. In this actions, we are going to use only 3 type of generation method. Basically, the small rotating drum, the Venturi setup, and the fluidizer, as seen here. And we are going to use also morphological characterization using electron microscopy, basically SEM or TEM. And this will help us to do measurement of mean diameter and length of objects. Also, we're doing some counting of these objects, the classification of this object, whether they are non-fibrous, fibrous, or fibrous type matching the WHO criteria. And that will help us to globally to have a concentration, number-based concentration element that will be used for a number-based dustiness element result. So this is going on. We started to have to send a call throughout the world. In a way, we had 14 partners that responded on this call. They are listed here, and the type of powders that we are going to study basically some carbon nanotube type of things. That's so for some fibers, and the silicon carbide type of things here. So basically, we hope to carry out all this work on the ARN within the 2022. So we'll be ready to have data and to perform and to do the writing of the TG in 2023. In terms of exposure modeling, work is geared towards defining what is the emission source rate here, that I show the formula here on the right side here on the top. The source is basically depending on the dustiness index, either in mass or number here, multiplied by the flow rate of the powder that you're dealing with, and multiplied by a H factor, which is an energy factor, and multiplied by the level of control that is shown here. So basically, these approaches is going to be used for exposure modeling through codes like 1-box or 2-box type of modeling codes that would help us to help us to assess what is the concentration in a near field or far field in a workplace. So work also, this TGD will provide some guidance on how you estimate the H factor, the emission factor. Of course, if you have a system that have a higher energy input, it will produce a higher basically source and that will be used for the modeling. So data is being used now to assess this approach. The exposure modeling approach, the determination also of the H factor, its sensitivity is already studied, and also this approach here will be useful for either granulated materials or a high aspect ratio nanomaterial. So now, yes. Okay, sorry, I'm late. Okay.
Not yet. You have 5 minutes.
Oh, okay. I'm sorry. Okay. So now 47 cases have been studied, 20 cases with high quality data, namely using dustiness index based on the small rotating drum and continuous drop. Work on handling energy factor is currently done. And we hope that this work on exposure modeling for the GD will be performed in 2022, and expected final submission in 2024. In terms of ATEX, the GD, how do you use the data for safety, ATEX safety risk analysis? So basically, this would be useful for what we call the inherent safety by design principle, basically coupling occupational safety and ATEX safety. That modeling that we showed before be useful for describing different scenarios, accidental scenario, and calculating basically the concentration and comparing with these LEL, with what we call the level, lower limit of explosion limit level of the powder. For example, ATEX, what is it? Is you need to have a combustible dust in the suspension, and sufficient concentration. And here you see on the right side what it means. So if you have a dusty materials, combustible, you can really produce a big blast, and then this has to come into the picture for the to assure safety for the worker. So development here is done through the GD right now. So we explain what is how do you assess the combustible nanomaterial? How do you assess the lower limit explosion of a material? And how do you would use the source term that is for the modeling, and compare to these predicted concentration with these lower limit explosion to guarantee a safer workplace for the workers. So conclusion, the OECD project is well underway with data generation on dustiness and ATEX data. Data analysis also and exposure modeling are going on. We also make sure that all the data that is produced is being stored on the perennial portal, basically the BSCW portal, and they will be also stored in the eNanoMapper portal. This is important for a fair principle. Difficulty encounters, of course, we have delays because of the COVID, and we may have even more delays from what we see now. We also see some heterogeneities identified between standardized dust distance methods and analysis. So that's going on. So we try to solve this. Result should be ready for the writing of the TG and the GD in 2023. So basically, globally, work will be carried out in 2022, writing 2023, submission 2024. And here I show you the contact if you are interested in contacting us. So you have these contact emails. Next OECD meeting also that is prepared. That is prepared for the January 22nd. That we are planning to do that, and we all thank all the contributors on this project. Thank you all.
Excellent, thank you very much. And we have 3 minutes for questions, and we have 4 questions, so we probably won't get through all of them, but we'll get started. Arne would like to know if you'd also measure the aerodynamic diameter of airborne materials to facilitate deposition modeling.
Yeah, we do the airborne distribution, particle size distribution. Yeah, okay. Through the LP measurement. Yeah,
and Neil would like to know whether, when measuring dustiness, there's obviously different material requirements for different methods. Which method requires the least sample?
Yeah, indeed. Yeah, that's a good question, because you have The rotating drum is probably the one that is using the most of it. And the least of it is the fluidizer. This is a new method, the fluidizer that's developed that's developed by BAuA. And I don't recall really the amount, but it's in terms of grams, it's less than grams and stuff. So it's a least amount that's used. So yeah, that is important, especially for new materials. If we need a small amount and they're doing some ranking and this is important to have an instrument that had used a small amounts.
Yeah. There's a short follow-up for Neil, and then there's one from Fleming in the chat, which you can probably answer in the chat later, just for time. The follow-up for Neil is why do nanomaterials need a different method to other powders in terms of measuring dustiness?
I don't get.
Why would nanomaterials need a different method than other powders?
It's not really needed, but we have at that point here we have different standardized methods, and the idea is to harmonize all this through this project, and these we have different methods because they are relevant to different energy. The small rotating drum, the vortex shaker, and so on, they have different energy, and they mimic different energy that is used in processes, and that's a reason why we have different methods.
Excellent, thank you very much. There's a couple of more questions in the chat. If you can look at them while we go.
Okay, yeah, I'm not too familiar with this, but anyway, I'll look at it. Okay.
Okay.
Thank you.
Otherwise, I'll email them to you. Thank you very much.
Thanks, everyone.
And if you can stop sharing your screen, then
sure.
Jakob can get on next.
Yes. Can you hear me, everybody?
We can.
Okay.
How do I stop sharing here?
I can do it for you. There you go.
Okay, great. Thanks.
Jakob, all yours.
If you yes, I will try to share my screen.
Yeah, and it's not in display yet. Okay.
Let's see if this helps.
Yeah, you just have to change your instelling for visioning at the top. It's sharing the wrong screen, so if you, that's it.
Okay. Do I say?
Yeah, that one.
Boot up?
Yeah.
Sorry. Yeah. Okay.
All yours.
Yes, thank you, Klaus. You have a quite Scandinavian name, so you could also read the Danish.
I was imported before Brexit 25 years ago.
Okay. Okay. Well, thank you. This is task 1.6 on identification and quantification of surface chemistry and coatings on nano and microscale materials. And this session will show the results from the intra-laboratory comparison, which involved NRCWE and BFR. And this intra-laboratory comparison will be succeeded by an inter-laboratory comparison, an ILC, next year in 22. And this will be the main activity in NanoHarmony for this task. And I will encourage you to participate tomorrow at 12 until 2 o'clock in stream 2, where we will have a session to discuss the conditions of this interlaboratory comparison. I will go rather quickly through the first slides, which shows the importance of analyzing the surface chemistry. It's quite crucial to evaluating the hazard of these materials, since it will change the properties. And as for now, there are no adequate analytical guideline documents available for this. So this is what this task should help, because it should form the base future guidance document. And the way we do it is, which has been presented earlier by Jutta Tensnert, that we start with cost-efficient, rapid methods to analyze, for example, bulk properties of materials. It could be, for example, based on thermogravity analysis to see the mass loss of a particular compound, and then we would move to more specific methods, typically mass spectrometry techniques, to identify specific compounds, and at the final stage then to quantify, for example, the inorganic and organic content of these surface chemistry compounds. And this is illustrated in this slide with the decision tree, where we move from simple to more specific and quantitative techniques in the end. So what I would like to do is to go through 5 cases, 5 materials with which we have been tested in the intra-laboratory comparison, and they will be used in 2 stages in the coming inter-laboratory comparison. And for this purpose, we selected 5 materials with different properties in terms of inorganic and organic surface chemistry, bonding type, and also 5 compounds which could reflect the use of qualitative and quantitative analysis, but also the use of different instrumentation and methods for their analysis, sort of to reflect what we would meet in the real world. And these 5 compounds were a titanium compound with both inorganic and organic surface coating, a silver compound with a PVP polymer coating, a modified clay and nanoclay, which was partly ion exchange with quaternary ammonium compounds, and a calcium carbonate core with fatty acid modification, and finally a fullerene, which was covalently functionalized with side groups to separate it from typically fullerenes. So with this choice, we use different types of coatings and bonding type. And this is an overview of the different methods and instrumentation we used. At NRCWE, we had for quantitative analysis, for example, the XRF and the TGA-MS, whereas at BFR, typically ICP-MS were used for quantitative analysis. And then we had some common techniques for qualitative analysis. That was, for example, multi-TOF, direct infusion, electrospray ionization, MS, and GCMS. And then BFR also had some unique methods, TOF SIMS and pyrolysis GCMS, and these were used for the different compounds. Some of these techniques are quite versatile in their use, because in the decision tree, you remember that we start with the simple analysis for the bulk properties, and then we go to more specific methods, and then for quantification methods. But some of these techniques, like the ICP-MS, the XRF, and the TGA-MS, could actually be used in all 3 stages. And I will not go into detail for all 5 compounds, but I will show you some of the general findings and illustrate the power of the different techniques in the following. So for the titanium dioxide, you can see how we have the titanium dioxide core and the inorganic coating, and actually two different organic polymers coating on the outside, the dimethicone siloxane and the polyethylene glycol, PEG. So with the TGA-MS, we could quantify the mass concentration of the organic polymers. The identity was not revealed with this technique, but this could be revealed with, for example, molding. ICP-MS and XRF could both screen, identify aluminum as the inorganic coating, but these techniques could actually also quantify them. And pyrolysis GCMS could also identify the organic polymer. This was also possible with tough SIMS, which could actually reveal both inorganic and organic coatings in this sense. But it's important to mention that these techniques were perfect for some compounds and useless for others, and vice versa. So it is a general outcome of this intra-laboratory experiment that you have to have quite many different analytical techniques in order to analyze a broad range of nanomaterials and their surface chemistry. This is, again, for titanium dioxide, moldy tough, showing characteristic peak patterns of the different organic polymers. You can see how the separation between the peaks reveal the monomer or part of the monomer structure in the polymer. And this is for quantification. We see how ICP-MS nail the aluminum and titanium from the core. And we also see how TGMS, by looking at the mass loss, which is the green line here, shows the organic content. For this, the silver compound coated with the PVP polymer, we see a quite interesting result where the TGA-MS finds about 15% organic coating on the material, which is in agreement with the supplier's information. But we also see how if we use TGA-MS in an inert atmosphere, we can see that we come up with a fragmentation pattern which can be compared with normal MS library results. So by looking at the fragments at MC85 and 111, we actually identify the structure of the monomer in the organic polymer. So TGA not only measures sort of the mass loss, the overall mass loss. In this case, it actually also identifies the structure of the organic polymer. And other techniques like the tough SIMS also do that, and also molding. And in fact, also pyrolysis GCMS. So for these compounds, many of the methods were actually quite successful. Then with the organic clay, we find mass loss, and this reflects the amount of quaternary ammonium compounds coated on the nanoclay. This is also indicated by XRF, since XRF comes up with an elemental concentration, and the difference between that and 100% would yield the water content and the content of organic compounds. The actual identity of the quaternary ammonium compounds could then be identified using either direct infusion electrospray ionization LC MS, but also normal HPLC MS, where we separate the individual compound and are actually also able to quantify them.
5 minutes left, Jakob. Thank you.
Thank you, Klaus. Yes. Then we have the calcium carbonate, and this is an example of how analyzing the surface chemistry is not that trivial, because if we look at the organic coding using TGA-MS, we would see quite a big mass loss. But the thing is that if we dig further into the spectrum, we see that about 5% of the mass loss occurs at a medium temperature, and then as we go to 600 degrees, we have an even higher mass loss. But this mass loss is not attributed to the organic content. It is in fact attributed to calcium carbonate being transformed to calcium oxide, which decarboxylates. So you also need to be a chemist in order to interpret the results, and you cannot use them blindly, sort of. We identify the different fatty acids and methyl esters coded with the LC and GC methods. And finally, for the fullerene, we had the challenge in this case that the core was organic and the side group functionalization was also organic. So this was a challenge, for example, for the TGA-MS, but we could use inert and oxidative conditions during the analysis to remove the side groups and detect their weight percentage of the whole material without ruining the structure of the original inert fullerene structure. That was possible with TGA-MS. We could also get in some information with pyrolysis, GCMS, and finally by direct infusion, electrospray ionization MS, we could see a molecular ion of the functionalized fullerene, and when we applied tandem mass spectrometry to this compound, we would yield the original fullerene structure, which indicates that the side groups were torn off using electrospray ionization. So these were examples how we could use quite a versatile fleet of instrumentation to analyze the different compounds, and these will go as recommended instrumentation and methods for the future coming inter-laboratory comparison. And there are a lot of other details associated with this activity, which will be discussed tomorrow at from 12 to 2. So I encourage you again to take part in this session, stream 2 tomorrow. Thank you.
Excellent. Thank you very much. Perfectly on time too. Mary had her hand up first, and then there's a question in the chat from Neil.
Okay.
If you get time. Mary?
Yeah.
Thank you very much, Jakob, for this very impressive amount of work. But I'm wondering whether, well, it is important for us when we assess toxicity, not only the type of covering or ligands, whichever on the surface, but we would like to know as well their density, orientation, and the functional groups that will be available to interact with cells. Perhaps this aspect to be considered, that we can determine these parameters and not only the type of covering material.
Yes, and I think that's a topic we should take up tomorrow. The inter-laboratory comparison will look more on the identification of the different surface coating, but what you request are more subtle techniques that look closer on the surface and their properties. So they are actually not included in this inter-laboratory comparison, although they are very important for the toxicity, as you say.
Thank you.
Excellent, thank you. It's always good to save something to tomorrow. Neil's got a question, and then there's some more you'll have to look at in the chat. Neil's question was, there's a lot of methods here. What guides a producer through which techniques they'll need for their products? Are there tier 1 techniques that will guide them through to the more complex tier 2?
Yes, part of NanoHarmony is delivery 111, where we submit an analytical report which goes deeper into recommendation and analytical settings for these techniques. And we also submit standard operation procedures, SOPs, for all of the techniques we have shown here. Some more detailed than others. That is a natural thing, but these information will also be shared for the coming inter-comparison. So the participants will not start from scratch. They will have a recommendation.
Excellent. Good. Thank you very much.
Thank you. I will stop sharing.
Yeah, and then Keld can start sharing, and then I'll let you deal with the rest of your questions in the chat.
I will look into the chat. Thank you.
Thank you very much, Jakob.
Thank you.
Next speaker is Keld Jensen, who's going to talk about solubility and dissolution rate in water and biologically relevant materials, matrices. Yep, perfect, Keld, all yours. Mary, your hand is still up. And we can't see you, Keld, or hear you.
So now it should work. It's just everything moved around when I was.
Thank you very much. All yours.
Thank you. Thanks. So I'm going to talk about now about the work we do in task 1.5 in NanoHarmony, which is the scientific basis for a new guidance document on the determination of solubility and dissolution rate of engineered nanomaterials. The task is led by NRCWE, and the partners are BFR and BASF, who also participated in preparing this presentation for today. And then we have formal associated partners in the project, URMC, IIT, and Chris and Forth, covering at least some regional worldwide spread for input into the project. So just a reminder of the background and the motivation for doing this work. It basically levels back to the OSD test guidance 105, which is a method for dissolution testing or solubility testing of water, of materials in water or chemicals in water. And it was found by several groups and analysts that this method is really not adequate for determination of the solubility of nanomaterials, and also that you do not provide information on dissolution rate with this methodology. And there's a new regulatory request for testing solubility and dissolution rates in biologically relevant media, which is of course not covered by existing OECD methods and not OECD GG 105. And then there are different uses of the data. For example, in READS, where there's a need for data for registration, grouping, and read across, but could also be data needed for interpretation of toxicological test results, predictions of fate and biopersistence, and actually risk assessment predictions. And here we come into the complexity, as there was historically a need for data for dissolution data or solubility data in water, but we really want to use data also to understand what is happening in biological compartments. And naturally, these different compartments are somewhat different from pure water alone. So here's an example on where it fits into the regulatory scheme. Here we have a snapshot from the ECHA guidance for REACH, Appendix R1, where the solubility is mentioned under the behavior. So where do they go? And the text associated here is water solubility and dissolution rate in biological media, as I mentioned before. And the solubility mentioned in the graph actually means both solubility and dissolution rate. And if we go into the more deeper test requirements for nanoforms, then we see that there is currently in the documents not a specific protocol indicated. It's mentioned that testing of materials should be identified by its PhysChem characterization as being a nanoform, of course, and the testing strategy is depending on its solubility and dissolution potential in relevant biological fluids and testing media. So it's also said then, in general, the exposure route is determining what a relevant medium is. So this opens up the box, of course, for testing in a number of different media. So, and I think it's important to note here that the determinant factor is really the route of exposure. So it needs to be considered what pathways the material is going through. So this led actually the initiation of the revision of the REACH guidance, and also what happens overall in the world, and also information requirements in Canadian and American regulations. And what I also understand from Asia is that we established an OECD WNT project to determine the solubility and dissolution rate of nanomaterials in water and relevant synthetic biological media. And the idea is to develop a guidance document for assessing these parameters. So the objectives of the NanoHarmony activity here is to establish the scientific basis to support the ongoing OECD project. And we have 4 main tasks to conduct here: is to gather information from relevant stakeholders on methods, SOPs, to determine solubility and dissolution rate, and it should be suitable for nanomaterials, and then also data that can be used for documentation. And based on the results from the exercise in bullet 1, we will develop, or we're developing a guidance document for intra-laboratory comparison study, or a round robin, as some explain it. And then we will conduct the inter-laboratory comparison study in coordination with the OECD WNT project, and finally analyze and report on the results from the inter-laboratory comparison study, as well as conduct some analysis on the materials to understand whether there are some transformations ongoing in the systems, but also some reaction chemical modeling to assess this possible transformation and reprecipitation phenomena in the dissolution experiments. So we have conducted surveys here from the previous meeting to get some input into to the suite of methods. We have mainly received from research projects, or actually alone from research projects, and then we have started working on the results to develop the guidance document for inter-laboratory comparison, and then start up the slowly the process towards the call for inter-laboratory comparison. So the main contributing projects we have received information from so far is from Gracious, Patrols, and Gov4Nano, where we have currently a parallel activity in Gov4Nano. Gracious and Patrols are finished projects where methods and data have been delivered, further investigation into contextual test parameters. In Gov4Nano, this is continued strategically based on knowledge gaps, and then there's been further validation of the a method for gastrointestinal dissolution testing, and one for a batch reactor analysis that I will show in the next slide. And then we have contributions from national projects, South Korea, Germany, Denmark, and I expect also from the USA. I would say here that it's possible still to come in last second with additional methods that can be considered in particular in the inter-laboratory comparison that we will call out for. So where are we now? We have 3 methods validated for testing dissolution of nanomaterials. One is the atmosphere temperature and pH controlled stirred batch reactor, which we have developed and implemented here at NRCWE. And this method is used to simulate initial to long-term solubility, as well as water and biodissolution, and can also be applied to understand the dissolution rates in different media. And here we work with long-lining fluid simulant, the Gamble solution, and phagolysosomal fluids, which are respectively at pH 7 and pH 4.5. And then the next step here, well, it's actually ongoing. We also test dissolution and solubility in water. And here there's some similarity with the OECD guidance document 29 method, but it's stricter and then fitted for nanomaterial assessments, which is covered by 3 kilodalton filtration of all samples that are collected from the system. Another system is led by BASF. It's the continuous flow system. Several publications have already been made on this method. It simulates, in this application, pulmonary uptake, and we can study hours to years of biodissolution in this approach. And it's also applicable for different fluids. Gamble solution and phagolysosomal fluids have been tested so far, and also environmental waters, and should also in principle be applicable for dissolution testing and solubility in water alone, but that would require some adjustment if we want to turn the method into also analyzing solubility. This method implements a method that was described in ISO technical report 19057. And the last method is a sequential gastrointestinal dissolution method that is led by BFR. It simulates biodissolution in the gastrointestinal tract by passing the exposing the media in the nanomaterials in 3 different media, starting from saliva, going into the stomach, and finally intestinal. And there's a specific sequence to go through this, and the material is in that way passing through pH 6.5, pH 2, and then finally pH 7.5 with different time steps. This approach is a modification of the German standard 19738, and would also agree with requirements, for example, in the EFSA guideline for food and food packaging. I will not go much into details here, but I'll just show you an example of the differences you get in solubility when you use different methods. And here you see for synthetic amorphous silica materials, quartz, zinc oxide materials, barium sulfate, and serum oxide. And clearly you can see quite different levels of solubility in the different experiments done here in, for example, blue and orange, the 24-hour dissolution in phagolysosomal fluid versus the Gamble solution. And here we have in gray the 5 minutes dissolution in saliva and stomach fluid for 2 hours, and then again saliva, stomach, and intestine for 2 hours. You see that the silica materials dissolve quite a bit in the Gamble solutions, and to a lesser extent in the gastrointestinal system, as well as in the phagolysosomal fluids over here. And quartz, as expected, lower solubility than observed for the amorphous material. And in fact, it's surprisingly high and probably associated with the presence of some organic, amorphous silica in this DQ12 sample. Then we move into the zinc oxides, and we see a somewhat similar pattern across all 3 types of nanomaterials. Here we have uncoated nanozinc and coated nanosink and a large nano/small micro-size zinc oxide here. We see the same pattern, very high dissolution in the fecal lysosomal fluid, to some extent in the lung lining fluid, and then variable solubility in the gastrointestinal fluids. And then we move to the barium sulfates and serum oxides, which have relatively low solubilities in these media, and least for the serum oxide. So this illustrates the importance of the different methods. You come with different results, and it's quite evident that you cannot do a direct read across from one compartment to the other without knowing a little bit about the behavior of the different materials in the different compartments. So I would say this example illustrates the importance of the dissolution testing and solubility testing in the biologically relevant simulant fluids.
5 minutes.
Yes, thank you. As I said, we would like to have some further contributions. In particular, we're talking about the whether some people have actually modified the TG105 methods to be applicable for nanomaterials, and we would welcome those for the inter-laboratory comparison. We are now in the last stage working on a SOP for the flask method and the stirred batch reactor methods to mimic basically the flask method in the TG105, because we have a specific requirement to also enable solubility testing in water, as also requested from regulations. This would require some modifications that we are still discussing, but of course the ultracentrifuge filtration is definitely needed as part of the exercise. We're also investigating the importance for different test parameters for the guidance and the SOPs, in particular done in the past and ongoing projects. So we've been looking into the effect of pre-dispersion, whether you get significant dissolution at the start during this dispersion process, which is needed for the batch reactor methods, the effect of the agglomeration level. To some extent, high levels of agglomeration could slow down dissolution of nanomaterials, at least from a theoretical perspective. For the continuous flow methods, there's been a study on to find the optimum flow rate for doing the dissolution testing, because you risk over and under saturation, and we would like to have a system that is adequate and mimic dissolution in vivo. And for all methods, we are looking into the composition of the test media, role of pH control. In particular for the batch reactor methods, we are looking into the effect of test atmosphere, whether it should be controlled or can be done in air, and then the filtration issues that is also discussed in task 1.8 yesterday, for example. So just an example on the effect of pre-dispersion. Here we looked into the use of different approaches, because different approaches have been discussed and proposed. So one is to disperse directly in the test media, so gamble solution, or water alone, or a BSA water, which is used in our standard method, which is the so-called nanogenotox dispersion protocol, which is basing dispersion on ethanol pre-wetting and a little bit of BSA in the water. And we have seen that in some cases we can increase solubility over time if we use the BSA water approach, and in other cases we delay dissolution, for example here in the fecal lysosomal fluid, where we see immediate dissolution of NM111, for example, and slower kinetics for dissolution in the Gamble solution, which is in part related to the pH aspects. And we did that for a number of different materials, and we conclude that BSA sometimes slow down the initial dissolution, but in some cases also enhance the dissolution that we have studied over 24 hours in this experiment. And this could, to some extent, be related to better dispersion. Issues is also that proteins are present in biological systems and make this observation relevant for consideration. And we need to understand better in the future how we or the influence of this, so far it looks like it has mostly influence on the initial dissolution rates, and of course this needs to be considered. Here's an example of the effects on flow rate on dissolution rate in the continuous flow system, and using this method and a range of materials, Keld et al. Found that they could identify an optimum where the biodissolution was mimicking the biological observations, and from that approach identified the optimal dissolution, or sorry, flow rate, considering the surface area of the nanomaterials. So this is also something to be used in the guidance coming out, and the SOP is, of course. So the timeline for the work and remaining work is that we are now meeting with experts and preparing the guidance for the inter-laboratory comparison. We will start calling out for contributors, binding commitments to the inter-laboratory comparison here in December, and then have a number of meetings with the inter-laboratory comparison partners in January to March, to get the methods up and running, and of course starting testing as soon as possible. We have an aim to try and finish the inter-laboratory comparison over 7 months, and then, because we would like to see if we can keep the timeline to get a proposal into for a guidance document for the OECD in November 2022. It's ambitious, and we hope that we will be able to actually do this and not hindered by another surge of global pandemic. So thank you very much for your attention, and there's a little time for questions maybe.
Excellent, thank you. Yeah, we got time for one question. The one that was first in the chat was from Astrid. Why was stirring selected instead of shaking the A-temp SBR?
For this setup, to you know, equip it with the electrode and monitoring that we do, then I think the stirring approach is needed. We are not at this point in time closing down the shaker systems, so these will be invited as part of the inter-laboratory comparison as well, because part of the inter-laboratory comparison is also to compare different methods. As we are aiming for a guidance document, we try to see the comparability between the methods as well. So it's not a lockdown for alternatives.
Excellent, thank you. If it's a quick one, Mary, then go ahead. Otherwise, I'll ask you to put it in the chat.
It's very quick.
Good.
Thanks, Keld, for the presentation. You mentioned filtration and all that. I'm just wondering, in your static, when you did you include or considered using the membranes rather than later on to filter the product to separate ionic to the particulate one?
Yes, we did consider that, and it's from analyzing literature and some experiments, it's not recommended to use these membranes due to a slower diffusion of ions across the membrane and some uncertainties in the dispersibility of the materials within the membrane. It's something that is already described in guidance document 318. But again, it's up to the test to see whether if some people come in with this method and to see how well it compares with alternative or the other methods that we put up here, for example, the filtration.
Yeah. Sorry, Klaus, quickly.
Thank you very much. There's plenty more.
Yeah, because filtration will produce other artifacts as well. That's my concern. Yeah, okay. Thank you.
There will be more time to sorry to cut it short. There'll be more time for this tomorrow. There's an expert group on this task and the one before tomorrow. But if we want any coffee, I'm going to have to say thank you to Keld and welcome to Ilse.
Thanks a lot.
Okay. Ilse is going to talk to us about the toxicokinetics of nanoparticles, and I'll leave it there with you, Ilse. Thank you for sharing.
Yeah, so just checking if you can all see my screen and hear me, because
Sorry, I muted you instead of unmuting me. We could hear you. So you just need to unmute. You need to unmute, Ilse. Ilse, I've managed to mute you. There you go.
Okay.
Thank you.
Thanks. Welcome. Good day to everyone. I realize that there might be a new face to some of the participants here as I joined the project 1st of January this year. And I would like to give you an update on task 1.3, the toxicokinetics of nanoparticles today. So as you know, the work done in NanoHarmony is supporting the OECD project, where a new test guideline on toxicokinetic studies is being developed to accommodate the testing of nanoparticles, since the current test guideline 417 for chemicals was found not to be suitable for nanomaterials. And this OECD project is on the lead of the Netherlands and the UK. The objective of the NanoHarmony task is to provide a scientific basis for the minimum requirements of the study design of in vivo toxicokinetic studies. And these involve in vivo studies following the oral route as well as the inhalation route. And we hope to give advice on what dose levels and ranges should be chosen in this study design, what should be the duration of the exposure and the post-exposure period, the time points for determining organ burdens, as well as the key organs, tissues, and excreta that need to be analyzed. The approach we take here is that we would like to apply kinetic modeling, as well as using available data from already conducted toxicokinetic studies, and also perform some new studies as well as identify studies that are currently carried out elsewhere. And we focus on nanoparticles with a slow dissolution rate or a moderately quick dissolution rate in relevant physiological media. I would like to thank the partners in this task, UK Health Safety Agency, NRCWE, BASF, as well as the associate partners here, and ENSO and NFIDS for their contribution to this update of today and their contribution to the task so far. To explain to you where we are with the activities, I've highlighted the timeline here. We're currently in the gray box that is enlarged on top. And we are after the SPSF project proposal was officially approved by WNT. The activities of NanoHarmony task are given in blue here. We had a workshop last year where we discussed the approach for the gap analysis, and this resulted in a deliverable report. And we are currently here at the second project workshop. There's another one planned in summer 2022. This will focus more on the newly generated data, and it all should finalize in the scientific report that is due in March 2023. Below, I've highlighted the activities in the OECD, and to see also where the associations lie. So after this second project workshop on the 7th of December, there will be a joint WNT, WPM and expert working group held, where we will give an update on the test guideline development, and we will focus on the regulatory requirements and that also link to the purpose of a toxicokinetic study. In the last year, we have held monthly telephone conferences with the task partners, where we discussed the results of the gap analysis and prepared for new studies, discussed these study designs, and kept each other up to date with regular updates of the ongoing studies. and we also had some guest speakers from future associate partner list or associate partner list and other tasks to inform us. We've also been in touch with eNanoMapper, as we will be trying to prepare a data template for inhalation biodistribution studies, and if possible to extend this also for our distribution studies of different kinetic study designs. There are 3 deliverables in this task, and the first one I mentioned already was the report on the data gap analysis, which was delivered in time. I will show you in more detail what is in that report later. And the next one is a report on the new experimental data to complement existing studies, which is due in September 22. And eventually there's the scientific document to support the OECD activities in March 2023. So the goal of the gap analysis was to identify what existing information could be used for a test guideline on toxicokinetics, if this was sufficient information and what additional information is needed. And the approach taken was that for 3 model nanomaterials, this information has been gathered and analyzed. And we've chosen nanosized cerium dioxide and titanium dioxide as a model nanomaterial of a slow dissolving nanoparticle, and nanosized silicon dioxide as a model for moderately quick dissolution. The conclusion for these model nanomaterials was that for inhalation, we expect to have sufficient information from existing data, plus some of the new data that is being generated, together with kinetic modeling to roughly estimate the minimum study design requirements. For the oral studies, there were much less, and there we are uncertain if a combination of data would be sufficient for to derive minimum requirements. We also consulted experts during the previous NanoHarmony workshop, as well as in an OECD joint expert group meeting in December 22. And here are some of their conclusions and recommendations that are also written down in the gap analysis report. And we structured their comments based on, since we asked to identify gaps and also to prioritize them. And here we've listed what will be taken into account and addressed in the test guideline, what will be considered, and what has lower priority and what might need to be addressed elsewhere. As an example of what will be taken into account in the test guideline, it was suggested to have a separate requirements for the inhalation and oral exposure, so really make clear that there are 2 parts. And there was a need identified for clear definitions on dissolution kinetics, as well as the limit of detection of the analytical method used, as well as the background concentration and some other important things. What we will consider is the influence of the retention in the delivery system, volume of oral gavage, whether or not IV data can be useful, and the feasibility with respect to having a total mass balance. What was prioritized as a lower priority for developing in the test guideline was to broaden the applicability to particles in general and nanoparticles with fast dissolution rates, as it is expected that these behave similar to known materials. Then the use of in vitro data was found very important, but it was felt that this needs to be addressed elsewhere, for example, in a guidance document, together with the relevance of in vivo toxicokinetic data in rats for human risk assessment. And please note that this was the prioritization made last year and it may be subject to change during the course of the project. With respect to new experimental data, we have currently this overview for studies following the inhalation route. Within the NanoHarmony task, RIVM is conducting a inhalation study with silicon dioxide, and we are just waiting for the organ burden assessments. And also within the task, BASF is planning a study with a polymer with a fluorescent label. And the other studies that are listed here have finished in the meantime, and we expect that we can use this data. With respect to the oral studies, NFIDS has just recently finished 2 studies with 2 types of silicon dioxide. And in the EU project Petrols, and has also been a study, an oral study where the cerium dioxide has been given in a snack versus gavage, and we have results from that. Also in Petrols, there was a study with food grade titanium dioxide that was finished, and there titanium was not above control levels. And we expect also this study with radio-labeled titanium dioxide that is performed by ANSTO that will start in January 2022. And I would just like to highlight here that if you are currently performing another one that's not in the list here or are planning one, it would be really great if you could let me know. We're all working, all this work will lead to a report on the scientific basis, and this is focused on the anticipated presence in tissues and the ability to detect nanoparticles. And this is also mentioned in the SPSF of the OCD project to depend on the dissolution kinetics in relevant physiological media. So that's basically the work that's related to what Keld has presented in task 1.5. And it also likely depends on the limit of detection and limit of quantification. And this is connected to the work done in task 1.4, led by Rachel. And therefore we have planned tomorrow a workshop, but this will first be in a smaller group of people just between the NH partners in this subsequent task to discuss the interrelationship. And yeah, what if there's also already something that we conclude here, how these how the solution kinetics and detection limits influence the toxicokinetics.
5 minutes, please.
Yes. This is my final slide with the outlook of the NanoHarmony task. It came after we finished the gap analysis report, it came to our attention that there might be data that's not publicly available, but still would be relevant to use. So we hope to get this a summary of this data to update the gap analysis report. And we also plan to take a closer look at the contribution of nanomaterial primary and agglomerate particle size to the biodistribution and limit of detection considerations for these 3 model nanomaterials, as this was not currently the focus in the gap analysis report. And we also plan to, for the kinetic in silico modeling, to prepare an overview that highlights, depending on the purpose of a toxicokinetic study, what potential in silico models could inform the toxicokinetic study design, and what are the state-of-the-art methods there. Yeah, so that was what I would like to present.
Excellent. Thank you very much, Ilse. Thanks for being perfectly on time. There's no questions in the chat yet, so we'll see if somebody puts their hand up or just start speaking. If there isn't any, or while people get one ready, I think it's quite impressive to see how many data sets you're able to pull in from other projects. Has it been difficult to get at the data, or are you getting it because the partners from NanoHarmony are directly involved in running them?
That's a great advantage here, actually. Yes, we can make separate data exchange agreements with the previous projects and with the partners. So yes, that really helps.
Excellent. That's good. Okay. Doesn't seem to be any other questions. I guess there'll be more discussion later. There's one from Richard Handy saying, for IV doses, nanomaterials do not spread evenly in the volume of distribution. Question mark.
Okay. Yeah, it may be due to my remark that IV data was mentioned might be useful in some cases, but we have to see in what cases we would actually be helpful. I think this is part of further discussion.
Excellent. Good. Of which I guess there will be more tomorrow. So good. Okay. Thank you very much. I'll take the opportunity to gain a little bit back of the time then, so we can have a longer coffee break. Thank you very much, Ilse. And Rachel, if you'd like to start sharing your slides and your camera. Excellent. Yeah, it's not in display mode yet.
Can you see and hear me?
Yeah, I can hear you and see you. The slides are not in display mode yet, for me at least.
Are they now?
No, they're in the speakers view. So you've got to go to display settings at the top and then duplicate or swap. That's it. Perfect. Great. Thank you. And I'll warn you when you're 10 minutes in.
Brilliant. Thanks. Well, morning, everybody. I do realise that I'm standing between everybody and their coffee, so I will try very hard not to overrun. So this morning I'm going to update you a bit on task 1.4 in NanoHarmony. This is led by the UK Health Security Agency with input support from the University of Plymouth and from NFA in Denmark. So just to go over some background. Task 1.4 is looking at the scientific basis for a guidance document on the determination of concentrations of engineered nanomaterials in biological samples in support of an OECD project with the same aim. This is included in NanoHarmony because a number of existing and proposed test guidelines have identified a requirement really to determine concentrations of nanomaterials in biological samples, but as yet there's no detailed guidance on how this should be undertaken. So there's a bit of a data gap. So we proposed the project and the activities that are being undertaken relate to looking at the state of the art and data gaps in this area. And we're also performing some experimental studies on single particle ICP-MS and enhanced dark field microscopy. And also, very importantly, we're holding various workshops to identify key data and obtain expert input, because as you appreciate, this is quite a large field. So just to reiterate, the methodology we've been adopting in task 1.4 is really sort of two-stage. So we have a data collection, analysis and interpretation stage, which is going to be ongoing throughout the project. We've undertaken an additional data capture phase to define the state of the art and identify gaps, although, as you'll hear, we'll be continuing with this throughout the project, looking at literature and projects, both national and international, and expert input. And we're generating some new data on single particle ICP-MS and enhanced dark field microscopy. The task 1.4 has two deliverables. Deliverable 1.8, which is a summary of the current state of the art and report on the data gaps analyses on these methods for determining concentrations of engineered nanomaterials and biological materials, and that was completed in March, and should be available soon publicly on the NanoHarmony website. And our final deliverable is a scientific document to support the OECD activities on the development of the guidance document, which is due in September of next year. I just wanted to briefly mention the scope of the OECD project. Clearly, it's focused on nanomaterials, but as we indicated in the SBSF, our primary focus will be on simple, non-complex manufactured nanoparticles, such as metallic or metallic oxide, as they're the most commonly in use and for which the most extensive development of techniques has been undertaken. But what we hope to do is to give some guidance on more complex and advanced materials to the extent that that is possible, given the state of development of the different fields. So we will try and look at multi-component and carbon-based materials and fibers. So one of the things we did when we were starting the project is do a very brief overview of the whole field. And what we identified was that single particle ICP-MS was what we considered a key technique to determine the concentration of nanoparticles within biological samples. There's a growing development of the technique and of use. And so we thought this was a very good starting point to undertake some assessment of the data and to consider the need for new experimental studies in this area. So we've undertaken a systematic literature review, and we've undertaken expert workshops, which were part of the first NanoHarmony International Workshop last year, which addressed things like extraction protocols and data gaps and measurement and validation. The results of our literature review have been submitted for publication, and we've just resubmitted after addressing some secondary comments. So hopefully that will be widely available soon. I know that many of you on the line will have attended our workshops last year, so I don't want to go through this in detail, but I just want to reiterate how useful these have been. At our workshops last year, we had over 25 experts from 9 European countries, plus South Africa, Australia, Korea, and USA, with experts from a wide range of institutions. And this was very helpful in developing our thinking. So this was our session B on validation, where we looked at things, we asked various questions on things like reference materials, acceptance criteria. On the left-hand side and the right-hand side, it is, I think, a very good summary of the conclusions from the debates and the discussions with the experts, which was very, very informative for us. So just to talk about a little bit about the next steps for the single particle ICP-MS work. On the left-hand side, you see a sort of a summary of our sort of conclusions from the literature review relating to sort of 3 different aspects: sort of research, development of guidance, and the production of reference materials. So in relation to research, what we identified was a need for more basic research on the effect of storage. That this is a key issue when you're undertaking various studies. There was also a need really to expand the range of nanomaterial and matrix combinations that have been addressed in the literature. There were some significant gaps. There's always lots of literature on silver nanoparticles, but for some of the other nanoparticles, there was a limited range of tissue matrices that had been considered. And also for the extraction protocol, there are very few sort of comparison exercises. So to address this sort of research area, starting in the new year at the University of Plymouth, we're going to undertake some targeted experimental work to look at the effects on samples of repeated freezing and thawing, storage time, freezing time, and collection tube types. We're hoping to expand the range of materials by looking at steel nanomaterials. I realize steel is not a new material, but it does have the advantages that it is multi-component. So that's what we're trying to look at, how single particle ICP-MS works for multi-component materials. And also we're currently considering undertaking a possible very limited inter-laboratory comparison exercise in next year, just to explore some protocol issues. Another thing we identified during the literature review is the need for there are issues with the development of guidance. Lots of issues around terminology, unsurprisingly, in that this is a newly sort of developing field. So consistent use of terminology, and also things like very important things like the systematic approach to the selection of protocols, and in particular the use of different types of controls at different stages in the process. So that will be input to the final task 4 deliverable, and we're hoping to discuss these issues further with experts, both through NanoHarmony and through the OECD expert group in the coming months. And a final issue that we discussed, which was a key factor from the workshops last year, was reference materials. There are very few, there are no absolutely sort of defined certified reference materials that can be used for single particle ICP-MS within biological samples. So starting in the new year, there'll be a project at the University of Plymouth to look at the viability of using some existing fish tissue certified reference materials as what we might call representative test materials for engineered nanomaterials. So I did mention that we had a focus on single particle ICP-MS, but we have been looking at other techniques. We undertook, as part of the first deliverable, a review of microscopy techniques. Clearly, electron microscopy techniques are widely used for the characterization of pristine nanomaterials, and various microscopy techniques such as Raman scattering, laser confocal have been used in many toxicity, ecotox, bioaccumulation studies to look at the localization of nanomaterials. But there are really very few examples of their use in a quantitative manner. So generally, there are two quantitative approaches to using microscopy quantitatively. One is counting objects in situ, i.e. in sort of biological sample sections, so appropriate sample selection, and using morphometric stereological approaches. There's lots of information on that, lots of guidance, but to date there are very limited examples in the literature of their use for nanomaterials. The alternative approach is digestion of the biological matrix and extraction of the particles, which are then mounted on a substrate for counting by microscopy. There are very limited examples, mainly in the literature, mainly for fibers, because this is an approach that has been used for decades for looking at asbestos in tissue samples. So clearly it's necessary also for the microscopy technique to be capable of resolving the individual particles. One of the difficulties with these approaches, especially electron microscopy, it can be used with a wide range of particles, but it is very time consuming. So this reference from Kempen from 2013 estimated that it would take over 400 million hours to analyze a single mouse liver using scanning transmission electron microscopy, which is clearly a little bit beyond most institutes' capabilities. So this really has implications for the levels of dose that may be required in any toxicity or ecotox study. But there are hopeful signs. So advanced automated image analysis systems could help in this area. And Jacques earlier this morning mentioned Fiber Detect, which BAuA have developed, and we are looking at whether that might be something that that in the future might have benefit for identification of particles within sort of biological samples. So our conclusions really on the microscopy side, whether there's a lack of practical guidance on the use of quantitative EN approaches in study design, in particular on dose levels, to enable judgments to be made on its usefulness in any particular situation. Other microscopy techniques may offer a better solution, for example, in that enhanced dark field microscopy and Raman's microscopy, and have been used in studies involving both metallic and carbon-based materials. However, the equipment is not widely available, and so more detailed consideration needs to be given to the practical use of these microscopy approaches before they can be more widely adopted for quantitative application in tox or ecotox tests. So just to show an example, this is an image from Mercer et al. in 2018 which indicates very clearly some carbon nanotube fibers, and showing that a dose producing 12,000 fibers in a microscopy section for EDM would only produce 6 fibers in a TEM section. So you can see the advantages of scale of these sort of microscopy techniques. So I also mentioned carbon-based engineered nanomaterials. At the workshop last year, there was a lot of interest shown in us trying to give some guidance in the guidance document on the assessment of carbon-based materials. Clearly it's a challenge, but is an area of increasing importance given the growing production of such materials. So we undertook a brief literature review in this area, again, to feed into our data gap analysis, building upon a few key papers on analytical approaches. Clearly, there are a range of established methods capable of characterizing pristine carbon nanomaterials. There are about 10 ISO standard documents available in this area. But the detection, characterization, and quantification of carbon-based nanomaterials within biological samples presents additional challenges, and there are examples in literature, but there are currently no international standard approaches in this area, although there is one ISO project currently looking at developing a technique. So there's a wide range of techniques with a wide range of reported detection limits, but the main conclusions of the review were that there's currently no established routine techniques or standard methods, and there's a lack of practical information for many of the techniques required for their routine application. And overall, we felt that probably the establishment of standard internationally agreed methods suitable within for the OECD guidelines needs to be within a sort of regulatory context. And this is probably some years off, but that doesn't mean that we shouldn't try and give as much guidance as possible at this early stage.
Sorry, I missed 5 minutes. It's 3 minutes, Rachel. Sorry. You're so quick between slides.
That's okay. So one of the things that we are going to do in relation to the carbon-based nanomaterials is that we are having a workshop tomorrow called the Determination of Carbon-Based Nanomaterials in Biological Samples, the Way Forward for Regulatory Purposes. And we're very fortunate to have 3 excellent speakers who are going to provide a background, and then hopefully we're going to have a focused discussion around issues like what are the best sort of techniques to use given the need for robust measurements, but also required sensitivity. So if anybody's interested, we have quite a few participants, but if anybody else is interested, please just get in touch with me and I'll see if we can fit you in for that workshop tomorrow. So these are some of the things we're going to be looking at, things like the expected detection limits for techniques, whether these are developing over time, and all this also the availability of quantification techniques. So I've just got a last few slides. So this is just showing where we are within the OECD project. So as you can see from the bottom row, this is the OECD project. And we were very fortunate that we submitted our SBSF last November, and that was approved in April. So we've been at WNT since April with project 110, and we had our first joint expert group meeting in September, which was very useful. And I hope some people from that expert group will be on the line for the workshop tomorrow. So finally, what's the outlook? What are we going to be doing from now on? Well, on the left-hand side, you sort of see what we've been up to so far. So we've done a literature review for single particle ICP-MS. We've had a literature review for other measurement techniques and the expert workshop. These have all fed into the data collection and gap analysis. We've also started to look at data from ongoing EU and other projects. And all this is fed into our first deliverable. We are going to carry on with data collection. This is a fast moving field and we want to keep up with the literature. So we will be keeping up with that, and also making more efforts to try and make sure that we haven't missed anything from the other international national projects. As I mentioned, we are undertaking some experimental studies in the coming year on single particle ICP-MS and enhanced dark field microscopy, which will be undertaken at NFA. We have our expert workshop on carbon-based materials tomorrow. And we're also going to be looking at other options to get some more guidance development, getting some more expert input to our guidance development and address in more detail issues like adequate use of controls and protocols. And all this will feed into our final deliverable, which will feed in obviously to the OECD project. So I think.
Oh, you muted yourself, Rachel.
Yeah, it's not. It's stopping. But the last slide is just saying thank you for listening. So okay. It stopped at just the right time. So I think that's. I hope that wasn't too much of a canter through, but there's quite a lot of information there.
No, it worked perfectly. Thank you. There's a couple of questions from Anna. One is, why use TEM as a quantitative method? Why not use it as qualitative and then back it up with parallel ICP-MS or the digested organ for the quantitative element?
I think that's an important issue that I didn't really have time to raise, but yes, one of the things that we are hoping to give some guidance on is use of combining techniques. Because I think, yes, one of the things, yeah, exactly, is to use things like ICP-MS with microscopy, maybe with EDX as well, to be able to prove that the particles you're seeing are the same sort of elemental components as the original nanomaterials. But yes, that is another aspect that we will be covering in the guidance.
Yeah, and a follow-up question from Anna is, have you developed protocols that look at the problems of the knife carrying over materials during sectioning, so soft tissues like the gastrointestinal tract?
We haven't started looking at those in detail, no. I think for microscopy there is quite a lot of guidance out there, and I think we're hoping to distill that. But I think we have to be realistic. We can't address all of these issues, and we're trying to focus on those most sort of relevant, most useful. But I do accept that there are lots of issues around microscopy, and a lot of those, like details for nanomaterials, have not really been explored in detail.
No. Okay, good. That's all clear from the chat, and there's nobody holding their hands up, so that should mean we get 10 minutes for coffee. Exactly. So I'd like to thank the speakers for all being on time. The 10 minutes we lost was at the start, so I guess that's mine or ours for setting the agenda without a gap to get everybody in. So please be back at 10 past and we'll get started with Isabella talking about intestinal fate determinations. Thank you very much for the morning.